Welcome to Enzymology. Let's start with the basics. What an enzyme actually is, how we discovered them, and the two main ways scientists classify them. A catalyst is any substance that alters the rate of reaction without undergoing any permanent chemical change itself. Enzymes, along with things like amino acids and metal ions, are common catalysts. But enzymes specifically are specialized proteins that accelerate chemical reactions. Many of the reactions our bodies relies on simply wouldn't proceed fast enough at normal body temperature and pH without these specialized protein catalysts doing the work. Our understanding of enzymes is built up over a century. In 1835, Berzelius studied the hydrolysis of starch by marc extract and first proposed the theory of enzyme catalysis. In 1878, Willy Kuhn coined the actual word enzyme from the Greek meaning in yeast. In 1907, Edward Buckner showed that a cell-free extract of yeast could still ferment sucrose into ethanol, and he named this active substance zymase. This work that won him the Nobel Prize. Sir Arthur Harden then showed in 1929 that zymase was actually a complex mixture of enzymes, not a single substance. And finally, in 1946, James Sumner and John Northrop crystallized urease and other gastrointestinal enzymes, proving conclusively that enzymes are proteins for which both shared that Nobel Prize. So, what actually happens during catalysis? An enzyme may become temporarily, even covalently, bound to the enzyme it's transforming. But at the end of the reaction, the enzyme returns to exactly its original form as the product is released. This is a crucial point. The enzyme does not change the equilibrium constant of the reaction, it simply increases the rate at which the reaction approaches that equilibrium. Four features define enzymes. First, they are biocatalysts synthesized by living cells. Second, they are protein in nature. Third, they are colloidal and thermolabile in character, meaning they are sensitive to heat and don't behave like a true solution. And fourth, they are highly specific in their action. Each enzyme is built to act on a particular substrate or a small family of related substrates. Enzymes can be classified in a couple of ways. The first is by location of action. Intracellular enzymes, also called metabolic enzymes, are synthesized within the cell and act within the cell. Extracellular enzymes, like our digestive enzymes, are synthesized inside the cell but secreted to act outside it. The second and most widely used classification is by the general type of reactions catalyzed. The IUBMB, the International Union of Biochemistry and Molecular Biology, appointed an enzyme commission in 1961, and this classification system has been in force since 1964, with modifications in 1972 and 1978. It sorts every enzyme into one of six classes. A handy memory cube for the order is the acronym OTHLIL or OTHLIL, which stands for oxidoreductases, transferases, hydrolases, lyases, isomerases, and ligases. Let's go through the six classes with examples. Oxidoreductases catalyze oxidation, reduction, or redox reactions, transferring hydrogen or adding oxygen. Examples include glucose oxidase, lactate dehydrogenase, and glucose 6-phosphate dehydrogenase. Transferases catalyze the transfer of functional group other than hydrogen between a donor molecule and an acceptor molecule. Examples include aminotransferases, hexose 6-phosphotransferase, and transmethylase. Hydrolases catalyze hydrolysis reactions. They cleave a bond and add water in the process. Examples include pepsin, lipase, cholinesterase, and urease. Lyases catalyze reactions specialized in the addition of water, ammonia, or carbon dioxide. but interestingly, without actually using water in the reaction the way hydrolases do. Examples include aldolase, fumarase, and histidase. Isomerases catalyze isomerization reactions. They can produce optical, geometric, or positional isomers of a substrate. Examples include triose phosphate isomerase, retinol isomerase, and phosphohexose isomerase. And finally, ligases. They catalyze synthetic reactions, joining two molecules together with the simultaneous hydrolysis of ATP, providing the energy. Examples include acetyl-CoA carboxylase and glutamine synthetase. So, to recap part 1, enzymes are biocatalysts, protein in nature, colloidal and thermolabile, and specific in their action. They can be classified by where they act into intracellular or extracellular enzymes, or by the type of reactions they catalyze, remembered with the mnemonic OTHLE.